All right, my name is Sarah Pennebaker, and today we're going to be talking about brain stimulation in zero gravity. So how to use TMS and MEG to determine the effects of gravity on brain function. So in the study we're going to look at today, TMS and EMG were used in concert to measure cortical excitability during periods of zero gravity induced by a parabolic flight. Researchers found that the resting motor threshold of participants, which is the intensity of TMS needed to produce effects in the periphery, decreased during zero gravity. We'll examine how the study was conducted, potential explanations, and limitations that lend this study to further investigation. But before taking a look at this study, we're going to start by examining the major techniques used in this study, along with the benefits, drawbacks, and recent advancements in those techniques. So transcranial magnetic stimulation is a non-invasive technique used to study and treat clinical conditions like depression. It operates by administering magnetic pulses from an electromagnetic coil pressed against the skull as an electrical current moves throughout that coil, kind of in the setup shown in the picture. So based on the coil type and positioning, TMS can target specific regions of the brain and is used in neuromodulation to stimulate specific cortical regions, though inhibition can also be induced by frequency administration to the target region. Repetitive TMS or rTMS is also used more in neuromodulation purposes, and non-repetitive TMS is more commonly used for investigation, kind of like the study we're going to look at today. Several major benefits exist for TMS that kind of give it an advantage over previous methods. So like EEG, TMS has excellent temporal resolution, kind of on the scale of milliseconds, and this gives it the ability to measure cortical activity on a biologically relevant time scale. So compared to things like fMRI, which really don't have that ability. Additionally, while EEG allows for similar temporal resolution to TMS, TMS can target a much smaller area because it operates by stimulating that area of the brain. Finally, compared to previous techniques with excellent temporality and targeting, TMS is temporary and non-invasive, which allows it to be used in a much larger population of humans. However, there are limitations that are frequently highlighted for TMS, one of which is that TMS stimulation sites are limited to those that can produce a peripheral response, like a site in the motor cortex. And TMS also cannot be used to infer cortical activity directly, so it can't measure action potentials and firing as a result of a technique, kind of like EEG is able to. Now, association with EMG has advanced TMS technique further, allowing for quantitative measurements of cortical excitability, which will be discussed further in association with the recent study. Specific modifications shown in this study also allow TMS to be taken out of the laboratory for the study of brain activity in novel environments, like those without gravity. One recent study used TMS to evaluate cortical functioning in zero gravity. While previous studies have established physiological effects of spaceflight, very few studies have been done to evaluate the effects of gravity on changes in brain function. So using these TMS helmets, researchers actually evaluated the intensity of TMS pulses necessary to activate the motor cortex enough to produce a response. So EMG, or electromyography, measures the muscle response to nerve stimulation, and it was used in this study in combination with TMS to evaluate factors like cortical excitability. Using EMG, researchers were able to determine the minimum power needed to move the thumb from a resting state as a result of a TMS pulse administered to the contralateral motor cortex of that thumb being stimulated. This measure is known as resting motor threshold, or RMT, and is frequently used in this study. So this figure shows some of the results of the study, just that in terms of resting motor threshold. The first two data points are actually on-Earth averages of RMT, taken as a baseline to compare to measurements taken on the day of experimentation. Now, threshold recordings were taken before and after flight and were shown to be equivalent to baseline measurements. Measurements were also taken during zero gravity portions of the parabolic flight and are shown as the center dip, so that fourth point, and you can see a very statistically significant average decrease in RMS to the scale of about 6.6 points. This is another figure from the study that showed all 10 participants of the study along with their pre-flight, zero gravity, and after-flight resting motor threshold. This decrease in resting motor state was actually present in every individual that participated in the study, not just at the average. It was talked about a lot in the discussion for this study, and it actually points to a physiological mechanism for RMT during zero gravity. And this was actually one of the helmets that were used in this study. So before a zero gravity study was conducted, the researchers developed a method for a portable TMS that could be used outside the laboratory. The helmets were custom made to each participant using a fiberglass casting of their head, and the coils snapped firmly into place. Because they didn't have to target these regions themselves, it made this method feasible for administrators of TMS without a lot of experience and could potentially be used for self-administration or for at-home administration of TMS. However, along with the benefits of this technique, TMS administered via helmet also creates additional confounding variables. For example, the helmet increases the amount of space between the skull and TMS coils, necessitating a greater degree of TMS stimulation to produce the desired EMG effects. And finally, we're going to talk about the limitations and future directions of this study. So the primary explanation given for the results of the study was that increases in cortical excitability caused a smaller intensity of TMS pulse to be necessary to stimulate those neurons and produce a motor response. As this study was able to examine only the output, the resting motor activation from TMS stimulation, several limitations and alternative explanations do exist. So one possibility discussed in the study was that the upward shift of the brain during that flight could actually reduce the space between the coil and the brain matter that was stimulated. This is because decreases in scalp cortex distance actually reduce the TMS intensity needed to stimulate regions of the brain. Another explanation is that zero gravity states may impact the periphery, actually changing the way those neurons fire, which would reduce the threshold needed to activate your musculoskeletal system. Even more factors such as changes in CSF flow or equipment changes during flight can contribute to changes in RMT in the study. As such, future experimentation does need to be conducted to determine the mechanism of the effect of zero gravity observed in the study. More studies should be completed to determine whether there are changes in cortical excitability, especially in areas outside of the motor cortex, and whether changes also occur in the spinal cord and the periphery, which were all confounding factors and other explanations for the results. So finally, similar studies should be conducted in this area and look at changes in other points of the flight. So were these changes in resting motor state actually because of the flight or because of the zero gravity state? That's something that this study wasn't able to show that future studies should look at. And that's all I have for you today. Thank you for joining in.